Hey everybody. So we've already learnt about mitosis and meiosis, and remember the acronym we use to remember it is ITMAT C. Well, that's the acronym for mitosis anyway. But remember, I highlighted that it's only prophase, metaphase, anaphase, and telophase that are actually mitosis, and the interphase and cytokinesis, they're like outside of mitosis, and they're a different part of the cell cycle. So what we're doing here is we're actually zooming out from just looking at mitosis to that whole cell cycle now, and seeing what else scientists have discovered about the way that cells reproduce. So you can see here in this diagram, most of the cell's life is the interphase. So from the instant that a cell is first made at the end of its parent's division, it's in its interphase. So it's doing its thing, like I explained last time. Whatever cell it is, it's performing its role in the body. So muscle cells are expanding and contracting, and cells in your stomach are producing enzymes, and the cell's doing whatever it needs to do to contribute to the body. Part of the interphase, though, you can see is, well, this interphase is split up into three separate phases. So there's the G1 phase, the S phase, and the G2 phase. So let's talk about each one of those and what happens at each stage. Okay, so firstly, when the cell's first made, it enters the first G phase, or the G1 phase, and G stands for gap. Originally, scientists thought that gap meant that nothing was happening in the cell, except now we know that the cell's completely active, all its processes are happening, it's just not doing anything that's related to replicating and dividing. So there's two gap phases. In the first one, or the G1 phase, the cell it's probably growing because it had just divided. And it's performing all its functions, like I said before. All right, and exactly the same thing happens in the G2 phase. But there's this S phase in between. So after the first G phase, we get into the S phase, which stands for synthesis. And that means the cell is it's copying its DNA. So obviously, to divide a cell, there needs to be two copies of every chromosome. And that's what happens in the synthesis phase. We'll get to that in a second. Then the cell, once its chromosomes have replicated, it then goes back into the second gap phase, where it goes back to doing its normal job, and it waits until it gets the signal from either outside the cell or inside the cell to then divide during the mitosis phase, or the mitotic phase. So that's what the M stands for here. So we've talked about ITMAT-C, but a better way to think of the cell cycle is that we've got G1, then we've got the S phase, then we've got the G2 phase, and then we've got the mitotic phase, or mitosis. And if this is a sex cell, then it's going to be meiosis happening there, obviously. So then if we relate it back to what we know, interphase includes the first three phases, G, S, and G2. And then mitosis and cytokinesis both happen in the M phase. All right, so the last thing is to look at what is actually happening when the DNA replicates. Well, in the next level, you'll learn exactly what enzymes are involved in copying a DNA strand, but for now, we need to understand how, or just the basic parts of the chromosome and what they look like when they divide and replicate. So some new words to describe chromosomes are centromere and chromosome arm, firstly. So every chromosome, it has a point somewhere along its length called the centromere, and this is a really densely packed region of DNA. And it's important because that's where the spindle fibers attach to when the cell undergoes mitosis. And we'll talk about that in a second as well. So that's the centromere. And then the next thing is either side of the centromere, we've got the chromosome arms. So in this chromosome, we've got a longer arm down the bottom, and we've also got a shorter chromosome arm at the top here, because the centromere isn't in the middle. But if you go and look at a karyotype, you'll see that the centromeres can range from being right near the end to all the way into the middle. So you can have chromosome arms that are the same length, or they can be different lengths, like in this one here. All right, so remember, we always need to remember that if you zoom into a chromosome, it's made of a single strand of DNA. So we're picturing our DNA strand. Of course, we know that there are proteins that the DNA is wrapped around, the histone proteins, but we're only concerned about DNA here. So when we get to the S phase, the cell needs to copy its DNA into two, and that means we go from our single DNA molecule to having two DNA molecules attached to each other side by side like this. All right, and this is where we get to that classic view of chromosomes looking like an X shape. But really what we're seeing with the X is we've got one chromosome on the left and the other chromosome on the right, and they're attached at the centromere right in the middle here. So you can see on this electron microscope picture, electron micrograph, we've got the centromere, and it's sticking together the two DNA strands. One of them's the original strand and one of them's the copy that is ready to pass into the daughter cell. Okay, so once we hit that stage, that's the end of the S phase. So then the cell, it has its replicated chromosomes, and now we call each of these strands of the new chromosome sister chromatids. So if you ever hear chromatids, that means we're specifically talking about the chromosome after it's copied its DNA, and it's now two DNA strands next to each other. So sister chromatids are the individual DNA molecules in a replicated chromosome. And it's these two sister chromatids that pull apart when we get to mitosis. So in the cell cycle, when the cell gets a signal to start mitosis, we head into M phase. We know all the steps of what happens in mitosis. Here's a quick graph or diagram to show us. So the chromosomes line up at metaphase. The spindle fibers come out. They attach to the centromere in the middle of the chromosome, and then they pull the two sister chromatids apart, and then the spindle fibers contract back to each end of the parent cell. So then Cytokinesis can chop the cell in half, and we've got our two new daughter cells. Okay. So then we get to our two new daughter cells, and each of them has just their one DNA strand in each of their chromosomes. All right, so that's the cell cycle. There's a lot more detail to learn, but that's the basics for now.